Welcome, everyone. Good morning, good afternoon, good evening, wherever you are. Before we get started for today's event, I am substituting for Dave Rice. Let's jump into today's event. Today, the topic for discussion is hyperpolarized NMR for complex mixture analysis. The workshop will be led by Professor Patrick Guerrero from University of Nantes. And he will be joined by two other panelists. I think at this point I will hand over to Patrick, and let's have a fun workshop.
Thanks a lot to the Ivan colleagues for the kind invitation. I'm very happy to chair this webinar today and also to give a talk. So I'm going to give a brief introduction and to introduce the different lectures that you're going, I hope, to enjoy. On the topic of hyperpolarized NMR for complex mixture analysis. So as you may know, complex mixtures are everywhere in the chemist life. They can be encountered in metabolomics, in reaction monitoring in particular, and they can be complex for different reasons because they can be constituted of a mixture of many molecules, but also in many different concentrations with a great chemical diversity, and these complex mixtures form a great challenge for the analytical chemist and in particular for the NMR spectroscopist. So dealing with complex mixtures in NMR is not so easy because the vast majority of NMR analyses are generally carried out by 1D proton NMR because it's simple, it's very straightforward, but the problem is that 1D NMR suffers from several drawbacks. It suffers from resolution problems, and we've been showing, I think in a previous Ivan webinar 2 years ago, that there were many solutions based on quantitative 2D NMR to deal with this complexity. Mixtures can also be complex because they react in time because their constitution is modified a long time. And today we are going to deal more specifically with a different kind of complexity, which is the sensitivity issues of NMR. So of course, the sensitivity issue of NMR is general. It's not only for complex mixtures, but it becomes even more complicated for complex mixtures, which are generally constituted with molecules with very diverse concentration, and analyzing complex mixtures is often trying to find a needle in a haystack from that point of view. And hyperpolarization offers many different approaches to deal with complex mixtures. And today we've chosen 3 complementary lectures on 3 complementary topics, illustrating how hyperpolarization can tackle such a great complexity. So I'm very happy to have 2 extraordinary co-panelists, Indrek Weiler from Tallinn, Kirsten Münemann from Kaiserslautern, and the 3 of us are going to speak approximately half an hour each on 3 different topics. I will start by talking about dissolution DNP for T13 NMR metabolomics at natural abundance. And then Indrek Weiler will provide a complementary view of a very different and complementary way of doing hyperpolarized analysis of biofluids, this time with power hydrogen, reaching unprecedented levels of sensitivity. And then we'll switch to a different kind of application, which is much more chemical and less biological, and also focused on process engineering, with Kirsten Münemann speaking about the use of Overhauser DNP for online NMR spectroscopy and for possible monitoring applications. So these are 3 very different topics, and I think it will really show the complementarity of hyperpolarized methods for complex mixture analysis. So I suggest that we keep most of the discussion for the end of the 3 talks, because I think there will be very complementary answers and different questions from the 3 of us. That being said, we agreed with the Ivan organizers that we can keep some time between each talk for 2 or 3 very specific questions. So I would encourage to ask specific questions at the end of each talk using the chat. We will monitor the chat. And to keep more general questions for the end of the webinar. So I'm going to start. about the topic of dissolution DNP for metabolomics at natural C13 abundance, trying to show you what we've been doing along these lines in the last 5 years mainly, and started a bit before. So in our group, what we are trying to do in general is that we are trying to develop NMR methods to improve peak separation. Sorry, I have a problem with with the Zoom interface that I'm trying to remove. So please give me just a second. Yeah, I'll try to manage with that. Sorry for the interruption. So in our group, we are mainly focusing on data acquisition methods to improve the quality of data at the acquisition stage. And over the last 10 years, we've been working a lot with the development of fast 2D NMR methods to improve peak separation. But more recently, we've been playing increasingly with hyperpolarization method to improve the sensitivity. And what I will try to show you in this talk is how we can basically do C13 NMR at natural abundance with the sensitivity of proton NMR and for metabolomics applications, including on clinical samples, actually. So as you may know, C13 NMR is a fantastic tool because it offers the possibility to solve many of the drawbacks of proton NMR. Because as you know, proton NMR for complex mixture analysis, is good because it's sensitive, it's high throughput, but it suffers from peak overlap. And on the other hand, you've got C13 NMR, which offers one of the best peak separations that one could expect. The problem is that C13 NMR is not sensitive at all, as you know, due to natural abundance and to the gamma of C13. And as a consequence, C13 NMR is a very low throughput technique. And basically, almost nobody is using C13 NMR for metabolomics and in general for complex mixture analysis of dilute samples. So the question we're trying to answer is, can we do C13 NMR with the sensitivity of proton NMR to benefit both from the advantages in terms of sensitivity and in terms of peak overlap? And the answer is yes, we can do that with hyperpolarization. And for that, we're using dissolution DNP, as you will see. So in general, generally speaking, the sensitivity of conventional NMR is very limited by the weak nuclear polarization and by the very small population difference between the nuclear spin states. And as you will hear today, there are different hyperpolarization methods that can make it possible to get rid of this problem and to generate hyperpolarized states that can lead to very intense NMR signals. And in our case, we are doing this with dynamic nuclear polarization. So DNP consists in transferring the polarization from unpaired electrons to nuclei. And that can yield very high sensitivity enhancement because electrons have a much higher gamma, much higher gyromagnetic ratio. And this process is particularly efficient in the solid state and at low temperatures. Well, actually, Kirsten will show you that it can also work in different conditions, but if you work in the solid state and at low temperatures, you can reach electronic polarizations that are close to unity and transfer this polarization to nuclei such as proton or C13. So the problem is that, of course, this is very efficient for solid state NMR, but if you want to do liquid state NMR, you need to use something to be able to detect NMR spectra on a liquid state sample. And this something is dissolution DNP. So maybe some of you have already heard of dissolution DNP. It's not something new. It's been invented by Jan Henrik Ardenkier Larsen 20 years ago. And dissolution DNP is represented here on a schematic view. It relies on a two-magnet approach where the sample is first placed in a first magnet, which is called here the polarizer, where the sample is placed at a very low temperature in liquid helium, reaching temperature which are close to 1 Kelvin. And under these conditions, the sample is in the solid state. Of course, unpaired electrons have been added into the sample, and the sample is irradiated by microwaves. And these microwaves can help to transfer the polarization from electrons to nuclei. This can be further improved by cross-polarization between nuclei, but I will not go too much into these details today. And in a few minutes, in a few tens of minutes, you can reach a hyperpolarized sample, which is solid. And this sample can then be dissolved quickly and transferred very quickly and pushed with helium gas under pressure into the NMR spectrometer, which is placed nearby. And then in that case, the sample arrives in the liquid state, and you can perform an extremely sensitive liquid state acquisition. And that can yield sensitivity enhancement of several orders of magnitude. But of course, one has to keep in mind that once the sample has come into the liquid state, this hyperpolarization is going to decrease as a function of the apparent T1. And it's also an irreversible process. Once this hyperpolarization has decreased, once you've been using this hyperpolarization, you cannot polarize the sample again. So it means that you can get very high signal, but in a single shot, and for nuclei that can, for which this hyperpolarization can survive during the transfer. So there are, of course, lots of technical challenges into doing this transfer as fast as possible. And also, it also means that this technique is going to be most promising for nuclei with long T1s. So it's actually the contrary of regular NMR, where in conventional quantitative NMR, for instance, we don't like nuclei with long T1s. Well, here we particularly like nuclei with long T1, and that's the case of C13, and in particular of quaternary C13, as you will see. So this technique is not new, but I think we've been the first one to explore its potential for C13 NMR at natural abundance in complex mixtures. And actually, so it's a story that we started 10 years ago together with Sami Janin when he was still in Lausanne. And more recently, 5 years ago, we had the chance to receive this instrument in Nantes, which is a prototype dissolution DNP polarizer, which is built by Brucker. It's a very strong collaboration with Jim Kempf and Dimitri Eshenko and Roberto Melzi at Brucker. And they have built this prototype that we have been installing in Nantes in 2019. And for 5 years until now, we've been exploring its potential for analytical chemistry and more specifically for the analysis of complex mixture at natural C13 abundance. So 10 years ago, actually, the result you have on the screen here is the first result we obtained on a complex mixture at natural abundance. This result at that time was obtained in Lausanne. And we were, I think, the first to show that you could obtain a C13 spectrum in a single scan on natural extracts at natural abundance. Here it was a tomato extract. You can see that the spectrum was focusing on the quaternary carbons here for the reasons I mentioned before. And you can see that in a single scan after 30 minutes of DNP, you obtain much more signal than you would obtain in 12 hours of signal averaging on a conventional spectrometer. So one of the key features to achieve this result was to rely on cross-polarization. I mentioned that before. Actually, we did previous attempts to do that without cross-polarization, with direct hyperpolarization from electron to C13. And it turned out that when the samples were too dilute, the C13 spins were also too dilute. And this polarization transfer directly from electrons to C13 did not work. And a key element to get this work was to incorporate the cross-polarization from proton to C13. As suggested by Sami Janin 10 years ago. So actually, we have a solid-state probe inside the polarizer where you can do cross-polarization from proton to C13. So it means that the polarization is first transferred from electron to proton and then from proton to C13. All this in the solid state, and you can reach a maximum polarization in a few tens of minutes. So at that time, it was 40 minutes. Now we are more at 15, 20 minutes, something like that. So with this fantastic tool, and when we received our prototype instrument in Nantes, we first tried to demonstrate that the analytical performance of this apparatus was good enough to be incorporated into a metabolomics workflow. And through a collaboration with Catherine Debord and Annick Moin in Bordeaux, we showed that we could use dissolution DNP for a very simple metabolomics-like study based on C13 NMR at natural abundance. And we chose a very simple biological model here where we discriminated tomato extract at two different ripening stages, red and green. So actually, there were only two extracts here that were pooled and then aliquoted, so there was no biological challenge, but we just wanted to show that the precision and the robustness of the analytical setting was sufficient to be incorporated into a metabolomics workflow. And the results were very good. We were able to separate the different samples as expected, and also we were able to identify markers of this separation. So there was no new biological information at that time, but we were very happy to show that in spite of a very complex experimental setting, we could yield very reliable analytical information. And then we wanted to move forward, so we worked like crazy. And this guy, Arnab Day, who was a brilliant postdoc in the team for 3 years, has been working like crazy in optimizing the performance of this experimental setting. He's been optimizing basically every parameter that could be optimized into the workflow, from polarization, sample preparation parameters, dissolution parameters, and NMR detection. And as you can see here, he was able to significantly improve the sensitivity of our setting. And this optimization, which was really very detailed analytical chemistry work, was really key to obtain a sensitivity that made it possible for us to tackle more real and more complex applications on biofluids. And then came Victor Ribet, a PhD student in the team who defended his thesis in December. And Victor was actually the first to show that we can record hyperpolarized C13 spectra on biofluids at natural abundance and in a single scan. So what you have on the screen here is a C13 spectrum of urine recorded in a single scan after 20 minutes of hyperpolarization. And I mean, I was very impressed when I saw this spectrum first, because it has lots of peaks. If you try to record the C13 spectrum at natural abundance on urine on a conventional spectrometer, even with 10 hours of acquisition, you will basically see nothing or almost nothing. And here you have so many peaks that it opens a whole field of application for this technique. So you can see here that some of the peaks were identified here through spiking. At that time, some were not identified. We'll speak about it later, but since it's a new technique, and since there are basically no or very few databases in C13 NMR for metabolites, well, we are facing identification challenges. But that's good news. It means that we have lots of signals. And the sample preparation here is very simple. It just consists in freeze drying urine to concentrate a little bit. But you can see that the result is very nice. So of course, a very important question that we also discussed in the team 2 years ago is the question of quantification. Well, maybe if I come back to my slide on the explanation of the principle of dissolution DNP here, what you have to keep in mind is that each signal has a response factor which is very different for different reasons. First, the level of polarization is not necessarily the same for all metabolites. And most importantly, the polarization decreases as a function of T1 when the sample is transferred. So it means that each peak has a different response factor here. So you should forget about direct absolute quantification. But the first thing is that you can absolutely do relative quantification, looking at relative variations between samples, and that we're going to see on applications to metabolomics in a minute. And actually, if you want to do absolute quantification, which is generally something that you want to do in complex mixtures, you can still do it, but you need to calibrate the signal response. And of course, it also means that the technique has to be extremely repeatable. If you do 5 times the experiment, you want 5 times the same result, and it needs to be also very linear. And this is what Victor has been showing. Victor has been showing that by using standard additions, so you may know that standard additions consist in spiking the sample with known amounts of the metabolites that you want to quantify. You you can spike several metabolites at the same time. And Victor has been showing that with such an impressive linearity and repeatability, look at the error bars here, they are very small. He was able to use his standard addition curve to determine absolute concentrations of metabolites in these urine samples. So you could tell me, well, that's good, but maybe these metabolites could also have been quantified by proton NMR. Well, actually, it's true in some cases. So for some metabolites, these are the green bars here. They could also be easily quantified by 1D NMR. And in that case, we could see that hyperpolarized C13 NMR, so the blue bars, provided the same results, which is good. It means that we have an excellent match between hyperpolarized C13 NMR and proton NMR for metabolites that can be quantified by proton NMR. But here there were also two metabolites that were not accessible by proton NMR due to very strong overlap. Even deconvolution did not work properly. And in that case, we were also able to quantify them by C13 NMR with hyperpolarization. So then we wanted to move one step forward and to try to apply this approach to a real clinical study. So we started a collaboration with colleagues at Nantes University Hospital, and they set up this small size because it's the first one that we are doing. This small size metabolomic study on patients and individuals with chronic kidney disease. So we had approximately 40 samples. Half of them were control, so healthy individuals, and half of them were patients with chronic kidney disease. So for all of them, we had urine samples, and these urine samples were analyzed in two ways. We analyzed them with our C13 NMR metabolomics workflow, as I mentioned before, and we also analyze them with conventional proton NMR metabolomics on a very standardized configuration, which is the IVDR configuration. So it was a collaboration with Claire Canet in Bruckner. And as you will see later, the IVDR data the proton NMR data were processed in two ways. They were processed with conventional bucketing, like in conventional NMR metabolomics, and they were also processed with deconvolution and pattern recognition approaches, which is the approach that Bruckner calls the IVDR method. And so we did all this, and then we did some classical statistical analysis, which are done in metabolomics, and then we try to compare the different methods. And also, I also have to mention that we prepared quality control samples for the DNP just to, so quality controls were samples that were obtained from pools of different urine samples that we analyzed at different stages of the analysis of the whole sample cohort, just to check the stability and the repeatability of the method. So this is a workflow that we used. So we started from freeze-dried urine that were prepared for DNP with the typical DNP sample preparation, analyzed with our prototype, and then processed with NMR ProcFlow, which is our favorite processing tool for NMR metabolomics. But very conventional processing consisting in bucketing the spectra, and then statistical analysis with also a very conventional tool, which is MetaboAnalyst. What you can see on the right here is the first principal component analysis, which was obtained on all these samples. The first thing you can see here is that there is a tendency towards separation between the two groups. So we'll compare on the next slide to conventional proton NMR metabolomics. And you can see the blue points here are the QC samples, and you expect them to be all at the same place, which is a marker of the stability of the technique. And we were very happy to have this. And then here you can see the results obtained with C13 hyperpolarized NMR compared with proton NMR profiling. So proton NMR data, not hyperpolarized, processed in the same way. And here on the right is PCA obtained with pattern recognition and deconvolution approaches. So basically the three PCA provide the same level of separation, which means that dissolution DNP aligns with the gold standard. And then we also perform supervised analysis in order to try to maximize the separation between groups and find markers of this separation. This is a conventional metabolomics approach. And if you're familiar with supervised analysis, so you know that the typical tool is PLSDA. And you may know that supervised analysis comes with indicators of the quality of separation. This R square, which is the goodness of the separation, the Q square, which is this predictive power of the statistical model. And what you can see here is that in terms of performance indicators, hyperpolarized NMR performs better than conventional proton NMR profiling. And this can be attributed to the better peak separation, which is obtained with C13 NMR. And it performs as good as the gold standard approach, which is based on pattern recognition, but which requires a priori knowledge on the sample and on the resonances. So we were very happy with this. It means that C13 hyperpolarized NMR at natural abundance has the potential to become a new elephant in the room in the field of metabolomics. We are not expecting it to replace conventional NMR metabolomics, of course, and also for other reasons that I will mention after, but I think it can be very complementary from proton NMR. And we went further than that, and we tried to annotate the NMR spectrum, the hyperpolarized C13 NMR spectrum, and to identify the signals which were responsible for this discrimination. And to make a long story short, we were very happy because there was a good complementarity between hyperpolarized C13 NMR and conventional proton NMR. And we were able to highlight biomarkers, metabolites of chronic kidney disease, that were not highlighted by proton NMR. And the reason why they were not highlighted by proton NMR metabolomics is that they were completely hidden under very strong peaks, which was not the case in C13 NMR. So I think this makes hyperpolarized C13 NMR quite promising for metabolomics. If we step back a little bit, there are things to discuss, and there are many things that remain to be improved until dissolution DNP can be more routinely applied for metabolomics. First, there are, let's say, very practical questions. The first one is the time and the throughput. As I mentioned before, with this setting, the experiment time was about 20 minutes per sample. So that's quite good in terms of throughput. So that's not very different from conventional proton NMR metabolomics. But the bad news is that with our prototype, we could reach a maximum of 8 samples per day. So you might wonder why this does not correspond to 20 minutes per sample. Well, simply because there are lots of manual steps to prepare the prototype for analysis and to clean it after analysis. That's also very tiring for the operator. So automation is needed. And with our prototype, we also had a huge helium cost because for each sample, liquid helium was needed to cool down the sample itself, approximately 300 euros per samples. So these drawbacks are going to be solved because the very good news is that we have removed a few months ago the prototype that I showed. And next summer, it's going to be replaced by a cryogen consumption-free and automated dissolution DNP instrument, which is built by Bruker, that will be delivered in the summer. So we are very much looking forward to use it. I also expect that the automation, of course, will increase the throughput. The fact that it's cryogen-free will considerably decrease the cost. And I also expect that it could even improve the stability and the repeatability of the analysis, of course, compared to a manual operation. And then we do have scientific challenges to solve. The first one is the one of peak assignment. It's a new technique, and well, actually, there are very few C13 NMR databases. So we will need to build our own databases. And we also need to be able to record 2D NMR spectra. And I'll say a few words about it in a second. And then there are also some discussions that we can have about concentration because contrary to what Indrek will show you, the concentration that we are able to reach are good, but they are of the same, the concentrations that we reach, the limits of detection that we reach with C13 NMR at natural abundance are roughly the same as the ones you would reach with conventional proton NMR without hyperpolarization. So it's already good because we benefit from the good resolution of C13 NMR, but it would be even better if we would be able to do dissolution DNP followed by proton NMR detection, because that would really lower the limit of detection, the absolute limit of detection. So we're also working on that. So I will not have time to discuss in detail these perspectives. I can just tell you that for 2D NMR, we're relying on single scan or ultra-fast 2D NMR, which is very compatible with the single scan character of dissolution DNP. So basically, we're using techniques to record a 2D spectrum in a single scan after dissolution. And this raises a number of fundamental questions, which are probably out of the scope of the talk today. And for the second perspectives, where I can show one slide that I had planned, we have developed a method where you can record in a single scan after a single dissolution DNP experiment, both a proton and a C13 hyperpolarized spectrum at the same time. And we are now able to start recording hyperpolarized proton spectrum and C13 spectra on biological extract at the same time. It's still at the proof of concept level, and this is thing that we will need to explore in the coming years. So I went a little bit fast in the end, but I want to leave to stay in time and to leave time for my co-panelists. So I just thank the wonderful team that we have in Nantes, also all the collaborators that I mentioned throughout the talk. Thank you for your attention. And so I'm done. I think we do have time for one or two technical questions if there are. So you can ask them in the chat. So there is one question from Krish asking how useful. So I'm reading the question because not everyone can read them. Maybe
I can,
yeah,
if you
sure,
want, I
sure.
can read it out. Yeah.
Oh, sure, sure.
I'm just, my question is related to something recently I was, I saw. How useful could this SSFP type of methodology, which is steady state magnetization, free precision magnetization, which is used significantly in imaging world, but how useful could that be? Is there a technical reason why we will not be able to use on a C13? Because it's being used extensively on the F19 of trace materials
Yeah.
by the Toronto group.
Yeah. Well, here, I mean, the honest answer is I don't know. We need to dig into it. I mean,
Okay.
I don't know. I don't know. I don't know if some of my co-panelists have an answer here. If that's the case, feel free to express yourself. But I don't know. I really need to dig into the subject to be able to answer this question.
No problem. No problem.
Yeah,
We can discuss later offline. Yeah. Thank you.
You're welcome. Is there any other question for the moment? There is one talk. So there was one question, sorry, from Stan asking, do you predict this DNP-based technology to replace standard C13 NMR spectroscopy of extracts? Well, actually, yes. Actually, yes. I really see it as a real alternative, especially for extracts. Because basically, I mean, especially when you want to analyze cohorts of samples in metabolomics, there is no way you can do it with conventional C13 NMR. So I think if you have one concentrated extract and you want to perform a series of analysis with 1D and 2D NMR, well, of course, you can always spend several days of experiment and continue to do it with conventional NMR. But I really think that for untargeted metabolomics, where you have to analyze 100 samples, 200 samples, maybe 1,000 samples, there is no way we'll be able to do it with conventional NMR. I mean, not easily. So I really think that for the analysis of extracts, this is one application where we can expect a lot from dissolution DNP. And I guess the challenge in the coming years will consist in evaluating the potential of dissolution DNP for a great variety of extracts and see what it can do, what it cannot do, and we have many projects in the pipeline with that to try and answer this question. So if there is no other question, you can keep on asking questions that we will take in the end after the two other lectures. I'm going to stop sharing screen and I'm going to give the floor to Indrek Weiler.
As always, give a word for our sponsors. First, MR Resources, they've been around for 39 years at this point, servicing the international NMR community and doing pretty much anything and everything that needs to be done with your NMR system. Magnet quench recovery, magnet and system moving and relocation, service contracts, reconditioned NMR systems, reconditioned magnets. And it's always very important for me to point out that they have absolutely a full supply of spare parts for everything that they sell. And they have engineering expertise that I dare say is second to none. Give some thought to doing business with MR Resources.
So yeah, thanks Patrick for inviting me over today. It's a pleasure to talk here, and also thanks to the organizers and the Ivan community. Being one still a laboratory that runs a very NMR instrument, I really appreciate this active community and support and discussions that you guys keep going. But yes, I'm here today to offer a complementary insight to what Patrick has just told you about turning reasonably unsensitive carbon detected magnetic resonance into a more applicable and useful NMR tool for metabolomic studies. So what we are doing here is in principle trying to address some of the similar questions, but for proton magnetic resonance spectroscopy. So in general, I'm also from Europe, as Patrick is, but I come from this small country in the northeastern corner of Europe, right west to this big dark gray country over here. And we are here a National Institute of Chemical Physics and Biophysics, which is the largest NMR institution in the Baltic states, meaning these three countries here in the Eastern Europe. And we're an institution that is active in different fields of biology, physics, both theoretical physics and experimental physics, as well as chemistry. In terms of NMR laboratory, we are the largest laboratory in the region with right now about, depending how you count, 6 or 7 different instruments, and also historically a hardware development center where we still have a good range of home-developed mass probes, a home-built cryo-mass probe that was running here about 10 years before a commercial alternative became available. And we still we maintain a capability to do NMR between 10K to well over 1000K. So it's there is some engineering experience here to allow us to build stuff to do things that otherwise might be quite challenging to accomplish. Right. But of course, my own team's research is in the introduction a bit very similar by motivation to what Patrick said, that we do NMR spectroscopy and we all love it because it's a robust, it's a non-destructive, non-specific and inherently quantitative technique. I mean, probably everyone in the audience knows all of that. And all of us will also know that it's actually not very sensitive. I mean, compared to just about any other modern instrumental analytical technique in the analytical chemistry lab, it is actually quite, I wouldn't say miserable, but not the most sensitive technique around. A few orders of magnitude behind several other popular detection techniques, especially in metabolomic studies. And as also covered in the previous talk, there is a very obvious physics reason why it is what it is. That said, I love this slide from a review paper about 10 years ago, where they actually show that this, as now already proved, the least sensitive techniques that you could apply to a biofluid like urine, it actually shows you signals, or is capable of detecting the largest number of metabolites in urine. So this shows that actually arguably the least sensitive technique might be one of the most beneficial ones. And this leads you to wonder that what if we could somehow make NMR detect even more of these signals? But of course, it's not actually that easy. If you look at the proton spectrum of urine, and over here we are looking at a proton spectrum of human urine detected at an 800 megahertz instrument. So in terms of spectral resolution, yes, nowadays you can improve a bit, but certainly not by an order of magnitude. What you have here is also that it's evident that we are detecting a massive number of signals. It's a very crowded spectrum. It is a spectrum with a very wide dynamic range of metabolite signals in it. And well, certain things we can resolve here, but actually the really, really interesting stuff is probably going on at and below of the carbon satellite intensity level of the most intense signals here. And you would bet that even if those signals that you are interested in down there, you could detect them, they are probably going to be flanked and overshadowed by the big signals here. So we have a lot of signals. The practical limit of detection is probably in the mid to high micromolar region. I'm saying practical, that if you really go for it, you can probably dig deeper. Yes, all of that can be alleviated by 2D detection, but this will again cost you a lot in experimental time and throughput. So a lot of the dilute and interesting bit you cannot see because of either the sensitivity or resolution, or probably both. And I'm arguing that if we're going to do something about it, we should actually think about both of the things, how to resolve the dilute signals from the rest of the metabolome and how to increase their NMR response. So what I'm going to show you on the rest of my slides is that how we do it by using some form of sample preparation usually, then a chemoselective parahydrogen hyperpolarized chemical machinery that allows us to detect enhanced NMR signals. So effectively, it is a chemoselective process that we are doing. So there will be a heavy metal iridium-based catalyst that you put into this biofluid-derived sample. A certain number of analytes, because it's chemoselective, it's only a certain number of analytes from the biofluid are capable of interacting with the iridium. So chemoselectively, NMR responses reporting the presence of these certain analytes will be hyperpolarized by parahydrogen hyperpolarization processes. So all of this thing, and this is actually very organic chemistry technical now, but bear with me because I believe this is the true essence of the technique. It is this iridium-based catalysts. These are the meat of this. This is what makes this magic happen. So it all works based on the iridium heterocyclic carbene catalyst that was introduced by the Duckett group more than 10 years ago. And this iridium main ligand on the iridium, in principle, one can change this to tune the properties and the chemoselectivity of this process. But of course, most of these ligands are poorly water soluble, which means that we need to change the biofluid water environment to an organic solvent somehow. Of course, we need parahydrogen, which is the source of hyperpolarization. But this is also a reason to remove water from our sample, because hydrogen is actually quite poorly soluble in water. Just about any organic solvent is much, much better. Then there are other ligands in the coordination sphere of the catalyst, and the nature of these ligands allows to tune the chemoselectivity, meaning what kind of analytes can today interact with our catalyst. And in principle, there are experiments where we also depend a bit on whether there will or will not be protons in certain positions of the analytes that we want to see. All right. So, and this is what we do is derived from the SABRE experiment that was originally published in 2009 by Simon Duckett, which is a method where you put that very same catalyst into a mixture. You introduce parahydrogen, you shake the sample outside of the superconducting magnet in low field, and then you drop the sample in the magnet. So what happens is that at low field, the para-hydrogen spin order will be converted into non-equilibrium magnetization on the analyte after it dissociates and comes off the catalyst. So in principle, you will hyperpolarize and detect these same analytes with their natural chemical shifts. But in principle, it works. What we have here is a Saber hyperpolarized proton spectrum with several responses visible here from hyperpolarized metabolites. These big signals here actually come from the catalyst itself. So we can say that there are a few signals here, some signals here that are actually metabolites. So yes, this is hyperpolarized, single scan, quick hyperpolarized proton NMR, but the number of responses is actually really not that impressive. You can certainly resolve and see much more by regular NMR. Although, yes, there are certain good Saber substrates that we can detect like this, but I argue that this is not the way to go to learn more about the biological fluid. So yes, this is fast and can be made to be quantitative by calibration methods, but the analyte coverage is actually poor, and we're still stuck to a one-dimensional NMR spectrum. So what we're doing is not Saber, but we do something that looks almost the same. It still uses para-hydrogen. It uses that very same iridium catalyst, but we are not waiting for the analyte to go away. Instead, we are detecting the chemical shifts of the two para-hydrogen-derived hydrides while they are stuck to iridium, and while the analyte is still stuck. And this basically now we are detecting each analyte or chemosensing them via the NMR detected signals of these hydrides. And as shown by the Marco Dessari group from the Netherlands and about 10 years ago, this is a good way of both detecting and resolving a reasonably complex mixture. And in this case, it becomes a really simple classical Pasadena type PHIP experiment. So this is a very good approximation to a two-spin AX type spin system where you put para-hydrogen, you only populate these two levels, which energy levels, which explains in a very straightforward way of from where and how the additional polarization and magnetization comes from. But more crucially, so this is how we get the more sensitivity out of this, but maybe more crucially, in a very complex biological mixture, we all know that our proton NMR spectrum is between 0 and 10 ppm chemical shift range. Turns out that these hydride signals that now report the presence of our metabolites, they resonate very, very far away at -20, -30 ppm proton. Which is somewhere where you usually do not have anything biological background going on. So we are enhancing the signals that we detect and also placing them somewhere far away from those high concentration metabolites that we're not particularly interested in anyway. And the way it works is that the NMR sample stays put in the magnet all the time. There will be a capillary for hydrogen bubbling in the sample. There is a home-built bubbling control system that under pulse program control controls the bubbling. So effectively what we do is that we replace the relaxation delay in a pulse sequence with para-hydrogen bubbling. And in general, the logic of NMR remains the same. There is no field cycling, no movement of the sample. And a lot of methodology development for pulse sequences like this is still happening actively in the Radboud University with Marco Dessari. And well, when we use that same urine sample that I showed before in the shake and drop Saber experiment, and we do this and detect the hydride chemical shift region. This is the response that we get. Every analyte that binds iridium gives rise to at least 2 doublets, meaning that if we count the number of doublets here and divide it by 2, we get a rough approximation of how many metabolites we are detecting. And this here, nicotinamide in this particular urine sample was about 15 micromolar in this urine. So if you look at the signal to noise here, we are safely above the limit of detection on 15 micromolar sample. So probably you can guess what these here are. So it's but it's still quite congested, the spectrum. So it can be resolved in a two-dimensional NMR spectrum. And one important point here is that, remember, we are detecting or chemosensing these analytes via these atoms here. It doesn't carry any normal NMR structural information, but it's not completely lost, because analytes with similar chemical structure tend to show up at similar diagonal lines on this kind of NMR spectrum. And if we zoom into this red box, you can see how many different complexes there actually are. Helps you to appreciate that how many different molecules we actually might be detecting. Although we have to bear in mind that each of these doublets is not a molecule. Some metabolites may give multiple different complexes. But anyway, we're still looking at several hundred of doublet responses here. So once we had this in check in a biofluid sample under control, we wanted to do something useful with it, basically to show that this can be a useful targeted metabolomics tool to measure something. So we figured that we will measure something that we know what the correct value is, but you cannot do it by normal NMR because of, well, the sensitivity and resolution reasons already covered. So we reasoned that nicotine can bind to iridium. Nicotine is a nicotine consumption and metabolism is very, very well known. We played around with different sample prep methods and devised one that gives a reasonably pure sample with nicotine and its metabolites easily visible here. And we calibrated them for quantification and performed a hyperpolarized NMR pharmacokinetic study. And turns out that the pharmacokinetic clearance curve, how fast this compound comes out of the smoker's body when a smoker stops smoking, the kinetics of this curve are exactly the expected value known for nicotine, but measured by other methods. And all of this curve happens at least an order of magnitude below the limit of detection of a cryoprobe 800 instrument. So with this, we did, I think, a quantitative targeted analysis with NMR by what you cannot do normally with NMR. And in this study, we showed a 30 nanomolar limit of detection for this method. All right. So while doing this, I mean, this sample prep was based on solid phase extraction, which effectively filters some of the analytes out of the sample. So we decided to try to do it without it. And we identified what, other than water, what are the urinary components that require us to do solid phase extraction and sample prep at all. Turned out it was ammonia. There is ammonia in urine. So we develop a different way of getting rid of ammonia. And then we could do, again, that same kind of experiment, but without solid phase extraction filtering of the catalyst, which means that now the iridium binding should be the sole limiting factor of how many signals we can detect. And again, if we look into a specified spectral region, we can see that when we have excluded solid phase extraction, we do get many more complexes to detect. But of course, unfortunately, we know very little of what they are. But at least we have a lot of signals, so it's something one could work with. But while doing this, we also saw that it doesn't matter whether we measure the same metabolite in the biofluid matrix or in neat solvent solution, the frequencies of it reproduce really, really well. So we can keep the matrix effects under check to a very good degree, meaning that you could build a database and apply a database to identify signals in a very complex mixture. So this is a very, very small database of adenosine derivatives in this case, which we could then superimpose to this urine spectrum and identify a certain number of signals from it. But of course, we were not at this stage interested in adenosine or that much. What we were really interested in was a collaboration with a cancer research group who wanted to study in very limited sample amounts the AMP, ADP, and ATP, the energy metabolites. So we proved here that we know where they are in the spectrum, and they can be detected with this methodology. And thought that how hard can it be? And this actually turned out to be hard enough to take at least a couple of years to figure it out. But at least now it's finally under review. So the trick is that there shouldn't be any ATP in plasma, but there is ATP in cells because they live. Red blood cells, the most abundant cells. If we do, and if we do it, this experiment now to a no SB involved to the cellular fraction of blood, we can compare it how the hyperpolarized hydride spectrum looks for urine and blood, and we can already on 1D hyperpolarized spectra see the signals for ATP here. This is the nicotinamide that is in pretty much everything living. So we always use this for normalization and referencing. But the trick here, why it took so much time, was that when we started, the red thing is ATP standard in a neat solvent. So we knew where it is, we knew how to do it. But on our very first spectra, which is the black spectra, it's a beautiful spectrum. Nicotine, we knew it has ATP in it, and a lot of it. Nicotinamide signals with our database overlap perfectly, but there is nothing where we would expect to see ATP. And when we dumped a lot of ATP into this, eventually something showed up, the yellow lines here with a very broad line shape and in a very weird and wrong place. So clearly something wrong was going on there. Otherwise, the spectrum was beautiful, but we couldn't see ATP, what we were after. So it turned out that and of course, we should have known it beforehand, but we are not biochemists. So ATP in its biological functional form is coordinated to magnesium. And magnesium attached to ATP inhibits it from coordinating to our spectrum. And in the end, once we had figured this out, the solution was actually reasonably straightforward. We can add a bit of EDTA to coordinate two valent ions from blood, and voila, we get to the spectrum where we can detect ATP and ADP from a blood fraction. So now we have moved from urine to blood, which I believe is metabologically and diagnostically possibly a more interesting spectrum. However, while we were doing this for a few years, we always knew that there were a few signals also, not in this -20 and a bit region, but closer to -30 ppm of the same hydride chemical shifts. And we didn't really, didn't know what it was and just lived with it for a while. But then a few years ago, the Sauri group showed that the signals at -28, they correspond to amino acids. And of course, there's plenty of amino acids in everything living. So we looked deeper and eventually managed to figure out that the signals closer to -30, they correspond to oligopeptides. So small, not small oligomers, almost small biopolymers. And this was actually a very interesting mechanistically of how this all works, because it turns out that those peptides, they start to coil up helically or in the catalyst and around the catalyst. But within a certain application range, small oligopeptides can be detected like this as well. So what we know by now is that this technology can be used to target and detect N-heterocycles in this spectral region, amino acids in this spectral region, and also oligopeptides in this spectral region. So, of course, we also dream of doing a metabolomics study. And in preparation for the first metabolomics studies, we wanted to demonstrate that we have all matrix effects under control. So it's not a metabolomic study yet, but we are here comparing all possible modifications of NS-TRIP methodology on 2 urine samples or a series of 2 urine types that are much more different from one another than is reasonable in any realistic metabolomic study. So we're now comparing urine from 2 species, human and dog. And what we see here is that even from 2 species, if there is a same metabolite present, it overlaps very, very well. Certainly, there are certain metabolites that are higher in one species or higher in the other species, but in general, both are mammals and largely consist of the same metabolites. So this means that you can go metabolomics with this. Also, the amino acids, they check out reasonably, not reasonably, but really well from different urines, different species, different matrix composition effects, they overlap. In human, we found a series of further signals here that are absolutely absent in dog. We don't know yet what these are, but it's a very interesting question. And also in those urines, some very weak peptide signals. But of course, you shouldn't be seeing much peptides in a urine sample anyway. Otherwise, unless the patient in question has a kidney disease, but both patients in question were very dear colleagues. So fortunately, we were happy to see very limited peptides in these samples. So with that, I claim that NH-PHIP has been demonstrated for a series of nitrogenous analytes that can be detected. Also amino acids, oligopeptides, amines pop up briefly in these 2 papers. And while we haven't touched this much ourselves, I need to point this out. I think it's very important that Tessari published 2 years ago the fact that you can make this complex chiral, in which case you will gain a possibility to resolve enantiomers of amino acids. So you can resolve DNL amino acids by this methodology. I think this is very important because there are very few methods that can do it for several amino acids at a time. So all in all, I claim that we are ready for targeted metabolomics, as shown by the nicotinic sample, because it's really tolerant of large matrix differences. We could be ready for untargeted or classification exercises. And there are reasonably good demonstrated sensitivity gains and limits of detection. And what we are actually doing ourselves right now is that our polarization now, the magnetization that we detect, doesn't come directly from the Zeeman interaction anymore. So we do not need a very powerful magnet for sensitivity. So we are now porting this methodology to slightly more sensible and affordable instruments. For instance, this is a practical study for an agricultural research group, where they only had access to a 300 megahertz instrument because agricultural university, they have access to what they can access. This is they are interested in profiling amino acids in raspberries. They're developing, doing high-tech science to develop different raspberry breeds. And it turns out that I've outlined the signals of proline here, but in 1D NMR on a 300 on these raspberry extracts, especially basically raspberry juice, you can probably detect all of these, but resolving all of them was 5 or 6 hour exercise by proton detected 2D magnetic resonance. So not really feasible. So we applied this NH-PHIP methodology to this raspberry juice, and turns out that we can resolve all the amino acids that they cared of by hyperpolarized NMR in a 5-minute 2D experiment. So, and over here, I think this is another point that over here, we are not using hyperpolarization to necessarily boost sensitivity. I mean, we have plenty of sensitivity here, but we are harnessing NH-PHIP's property of placing the signals of interest somewhere else. Proline has a lot of signals. Over here, each amino acid only has 2 doublets. So it becomes much more easy to resolve them. So you may want to do this hyperpolarized experiment not only for sensitivity issues, but also when you want to just comfortably resolve something and possibly in a lower field situation. So with that, I want to thank people who helped all of this to happen from the Institute and from the group, and also the organizations that have allowed us to have fun in the lab. And of course, I thank you all for being here and listening to me. Thanks.
Thanks a lot, Indrek, for staying perfectly in time. So if you have one or two technical questions to, well, technical, scientific, whatever questions to Indrek, please add them in the chat. Well, I do have one. Well, very impressive results, including new results that I had not seen before. When you consider doing metabolomic studies, one of the sensitive elements is the sensitivity of NMR to pH. Well, this is well known, but I was wondering if you were expecting an effect of pH on the hydroid signals that would be typically the same as in conventional NMR, or maybe less, or maybe worse.
The same reason as in conventional NMR apply. I mean, there will be a pH, probably a pH dependence on chemical shifts. But there is one more crucial thing. So most of these analytes that we detect are basic to a degree. And once you iridium itself, where the analyte needs to bind, this is a positive charge center. And if you protonate something, it won't electrostatically be able to bind. So we need to control pH also to have like a quality control over what can coordinate and bind to this iridium catalyst and what cannot, and also to make sure that they do it in a reproducible and reliable manner. So most of this biomixture work now we do with different organic buffers in place.
Okay, yeah. Thanks a lot. There is a question for you in the chat. Is there a limitation to the chemoselectivity of this method? And second question, would it be possible to detect catecholamines like adrenaline or noradrenaline in urine samples using this method?
Yes. The main limitation here is that what you want to see needs to be able to reversibly, or not necessarily reversibly or irreversibly, but it needs to be able to interact with this iridium catalyst. Looking from very far away, just about anything with an electron bear might do, but it's not as easy as that. Yes, we right now know of N-heterocycles, amino acids, oligopeptides, and a few more other things, amines as well. There are some literature hints about sulfur being able to coordinate, but the whole chemoselective scope and where this can be turned, I think the whole scope is not fully explored yet. So I have based on experience, I have some way of guessing what will or will not work, but the whole range of what can be made to work is still a bit of an unexplored territory.
Thanks a lot, Indrek. So in the interest of time, so we'll come back to final questions after the last talk, and in the interest of time, we are going to switch speakers, and now we are going to come back to DNP, but a different kind of DNP, and also a different kind of applications, much more chemical now. And so, Kirstine, if you want to share your screen.
We invite you to get to know Q1. Q1 is dedicated to developing NMR systems from 400 to 600, as well as full system upgrades from 300 to 600 as well. You can get a lot of features in a 2-channel NMR spectrometer at a great price. So we invite you to get to know more. Contact us at q1americas.com or info@q1americas.com by email. Thank you.
Thank you very much. So I will tell you something about Overhauser dynamic nuclear polarization, which might be useful to be for online NMR spectroscopy. And we are just starting a little bit in this field, so our results are not that yet perfect than what we have seen already. But what is good that Overhauser DNP can work directly in the liquid phase. So, and as the aim of the lab I'm heading is to do process monitoring and reaction monitoring by NMR. That's very, very good because most of the processes and reactions are taking place in the liquid phase. So, and the best way you can monitor them with an NMR spectrometer is to take just a sample loop out of it and via bypass setup, and then let your sample flow through your NMR spectrometer and return it back. And of course, the requirements would be to have the transfer time as short as possible to get a good temporal resolution, and also to have the volume in the bypass line as small as possible. Do not disturb the actual process. And of course, we all know that benchtop NMR spectrometers were developed more or less directly for that application because they are very tiny and they can fit into fume hoods, and therefore you can bring them very close to the processes or reaction, and therefore they are optimal to do that kind of NMR spectroscopy. However, the great advantage can turn into a disadvantage if you have a look on NMR under continuous flowing conditions, because the magnet is very tiny, and therefore the polarization length before your sample enters the NMR coil for detection, so the distance where polarization can build up is very short in this benchtop NMR magnet. So in our one is around 10 centimeter. And even if you go with protons with T1 times on the order of just a few seconds, you see that even with very moderate flow velocities, you lose a lot of your signal because the liquid travels too fast through the magnetic field to build its polarization up. And if you want to monitor faster reactions, that might be the flow regime you want to look at. And we thought that maybe ODNP can do the trick here, and it would be very beneficial if we could do that even on more insensitive nuclei like carbon-13. So the basic principle of ODNP is very, very much related to the principle of dissolution DNP. We also add free electrons to our samples, so unpaired electrons. And for that, we just put organic stable radicals in our sample, and they will transfer upon microwave irradiation at the lambda frequency of the electron spins their polarization to the surrounding protons or carbons or what you like. Because we are working here at room temperature, so we don't have the benefit of going to very low temperatures. So we have only the signal gain of the gyromagnetic ratios of the proton and the electron, but that's also something. So it's 660 for protons, and for carbon-13, the best you can have is 2060. Of course, life is almost different from theory. So what you can actually get is something around 170 or 200 maybe, because this temporal radical has 3 hyperfine lines and you can saturate only 1, and therefore you have to divide that number by by 3, and then you end up with something like around 200, and then you have some experimental imperfections, and then you get to that real numbers. Okay, so in our lab, we use a very, very simple setup because we want to have it small, because if the benchtop spectrometer can be very brought very close to the process and you add something very massive to it, of course, you are then back to high field NMR, let's say. So we decided to do our Overhauser DNP in a tiny Halbach magnet, which is sitting on top of our benchtop NMR magnet. And we do the ODNP experiment at 0.35 Tesla, which is X-band for the EPR spectroscopists, because there every microwave components are readily viable and they are not very expensive. And what we are then doing is then we hyperpolarize here, and then we just flow our liquid into our benchtop NMR for detection. One key ingredient of that setup is that we use not dissolved radicals, but immobilized radicals. And that has the reason that, of course, the radicals reduce the T1 times, and therefore we would lose hyperpolarization after building it up directly if the radicals are still present during our transfer step. So what we are doing is we immobilize the radicals in a fixed bed, and then they are only present for the ODNP. So here is the EPR resonator where we do microwave irradiation, and then here's a FRIT, and then we come out with a native hyperpolarized sample and can easily measure our NMR signal. And the reason why that ODNP works so great under continuous flow is that, of course, in the presence of the radicals, the T1 times are shortened dramatically, and therefore the polarization buildup is much, much faster than in conventional NMR. Okay, we use controlled porous glasses as a carrier for our radicals because they are chemically inert. And what we do see is that we can have here a sufficient radical loading if we choose controlled porous glasses with a not too high pore size. And what we are doing then to increase the number of binding sites dramatically inside the controlled porous glasses is we attach a polymer on top of it, which has something like a hyperbranched structure, and on that hyperbranched binding sites we put the radicals, and therefore we can have a high radical loading, and that is what we need to have for Overhauser DNP. Okay, now we have everything in place, and we can see what we get if we do a simple experiment on a mixture of acetonitrile and water. And here's what you get if you just let the mixture flow through your benchtop NMR spectrometer. So that's all thermally polarized, and you see the peak of water and the peak of acetonitrile, and you see they are gone if you increase the flow velocity. For the hyperpolarized spectra, you see that at each flow velocity you have almost the same intensity. Of course, the signal to noise is much better because you have enhanced your signal. So I think we can say that ODNP enables 1H NMR in the fast flow regime. We can go even faster, up to a flow velocity of nearly a meter per second, so that's really fast. So in thermally polarized NMR, you don't have any signal there anymore. So that's all fine. But as Patrick pointed out in his talk, if you hyperpolarize something and then maybe you want to quantify it in the end, at least the hyperpolarization step must be highly reproducible. And that, of course, we have tested also. So once we switch the microwave on and get our hyperpolarized signal, it's stable over time. So we hope that we have the right reproducibility to do something like online monitoring. Okay, would be even better if we could do that also with carbon-13 because of its greater chemical shift dispersion and all the advantages that carbon-13 has. And even here, it's also the problem with the long T1 times of the carbon-13 increases, of course, the problem with the drop of your polarization buildup. And therefore, carbon-13 under continuous flow is nearly impossible if you just run it through your spectrometer. And now I will try to show you that ODNP can solve this problem also. So it's the same setup, basically, and we want to measure on samples that are not enriched in carbon-13. And to do that, I didn't tell you before that all that flow path here is just a 250 micrometer capillary. And that's good enough for proton detection, but if you try to measure thermally polarized carbon-13 and that to measure your enhancement factors, you will have a hard day or 2 or 3 or something like that. Therefore, we have to increase our detection volume. And we did it by just doing here an expansion to almost 3 millimeter. And but what we didn't thought of, and we had to learn it the real hard way, is that if you come here with a fast flowing liquid, something like a jet will form. So that means that your sample volume is not filled completely with your hyperpolarized liquid, but here comes just a jet out of it, and here are still a lot of thermally polarized spins, and you have some backmixing and really, really bad flow dynamics that you obviously don't want to have. But we will see if we can do carbon-13 measurements with that. Because at the moment we are building something better, but it's not there yet. So if we speak of ODNP of heteronuclei, we have to have a look to the basic hyperfine mechanisms that are the origin of Overhauser DNPs. That means the hyperfine interaction of the electron and the nuclear spins. There are two mechanisms. These two are interacting with each other, so dipolar and scalar interactions. If you go for protons, you have only dipolar interactions, which gives you negative signal enhancements. But if you look for carbon-13 or other low gamma nuclei, they mostly give you positive enhancements, but in many, many molecules, there's still a huge dipolar interaction also present. And because these are of different signal sign, they can cancel each other. And in terms of hyperpolarization, it's really, really bad because you have a positive hyperpolarization and a negative one, and they just cancel out, and you end up with nothing in the end. So, so let's see if we can still do something with that. So that's now acetonitrile running through our setup. And here's the thermally polarized spectrum measured without flow, because if we just switch on the pumps, the whole signal will vanish, and it's accumulated with 256 scans. And that's a 1-scan Overhauser DNP spectrum under continuous flow. Of course, the signal to noise is very, very minor here, because the enhancement values are not that high, but at least you can do it with single scan acquisition under continuous flow. What is not that great with a direct carbon-13 DNP here is that it decreases with increasing flow velocity. and at some flow velocity, you are almost you almost lose your whole signal. But you can do a trick here, and you can transfer the polarization of the protons, which is also present at very high flow velocities, just via the J coupling. So you use something like an ENEP sequence to the carbon-13, and then you can do carbon-13 acquisition also at quite high flow velocities, up to 2 meters per second. It looks quite different if you choose a different molecule, which has a much better hyperfine interaction to your radical, and that's chloroform, which is known to have a almost pure scalar interaction to the temporal radical that we are using. And that's again the thermally polarized spectrum measured without flow with a lot of scans, and that's the ODNP spectrum with a single scan at a half a meter flow velocity, and you see that we get a very nice signal to noise compared to the thermal spectrum. So the enhancement is much better here. And what is also different here is that the scalar interaction increases with increasing flow velocities because it happens on a much faster time scale. So the build-up rates for the dipolar and the scalar interactions seems to be different, and we are now verifying that with different experiments. But I think that we can also say that ODNP enables carbon-13 NMR in the fast flow regime, because now you can do it what can't have been done before. Of course, if you do reaction monitoring, you want to measure mixtures and not the neat liquids, and that's a spectrum of a mixture of acetonitrile and chloroform. And you see that we can polarize both to an extent that they are measurable in single scan under continuous flow. So what I have shown you so far is that maybe we can overcome the problem of measuring NMR at high flow velocities. But if we want to do reaction or process monitoring, we really need to quantify our signals. And as we have heard before, hyperpolarization is always very, very great. But if you want to have that nice concentration over time profiles that we normally have in reaction monitoring, we can't use something like direct integration here, because the signal intensity does not only depend on the concentration of your sample, but on the hyperpolarization level. We will have a really, really hard time to measure all molecules and then calculate the enhancement with the hyperpolarization levels and then integrate. So that's nearly impossible. So we will also try to calibrate the signal that we get out of the ODNP. And here I show you again the proton measurements under continuous flow. So that's again the mixture of acetonitrile and water. And here is pure acetonitrile, and then here water is added until we have a pure water sample. These are thermally polarized spectra, and these are the spectra acquired with Overhauser DNP. And you see here that the signal enhancement for the water and the acetonitrile is very, very different. And of course, that means that now we can't, that now the signal integrants will not directly depend on the concentration. So if we would do just a normal calculation for the amount of acetonitrile, then we would do with normal quantitative NMR, we will end up with our points of acetonitrile lying over the ideal diagonal, which is the ground truth in that experiment. So, and that's just because the enhancement of acetonitrile is much higher than the enhancement of water. And we have developed a calibration function to fit that curve, and that's basically just a combination of two linear functions with a smoothing here in the middle, so one gets smoothed into the other, and weighting functions for the different linear functions to see if we can fit that behavior. And actually we can. So here are all the points of the acetonitrile, and they are fitted by our calibration function. And you see that the function is fitting the points very, very well. And then we did a leave one out analysis, which is quite common in data science. So you do that fit without one point, and then you calculate that point back. And you do that for every of these points. And then you can determine the relative deviation from the truth. And you see it here that we have the highest deviation was something like 3%. And that's quite okay for proton NMR under continuous flow in benchtop spectrometer in a 250 micrometer capillary. The situation is a little bit different if you change your sample to two molecules which have almost the same hyperpolarization. And that is, for example, acetonitrile and dioxane. They have almost the same level of hyperpolarization. Here's again pure acetonitrile, that's pure dioxane, and in the middle, in the mixtures, and these are the ODNP spectra. The enhancement here is very, very minor, but we can measure it under continuous flow, so that's the good news. And here the enhancement levels are almost the same. And therefore we can say here also that the signal integrals would directly reflect the concentration in your sample. And if you just calculate your ACN concentration, like you do with normal quantitative NMR, you see that the points are really, really close to the diagonal. And but we can apply our calibration function also in this on this sample, of course, and then we do that leave one out analysis, and the highest error we get here is around 6%, and that is because the hyperpolarization did not work that well in the sample, and therefore, of course, the integration is not as good as in highly enhanced spectra. Okay, now we will see if we can use that also for carbon-13. So that's again the mixture of acetonitrile and chloroform. Here is pure acetonitrile, and this is pure chloroform. And I can't show you the thermally polarized spectra here because we couldn't measure them, of course. But you see that we have a quite good signal to noise in the hyperpolarized spectra. And as we have known already, of course, the enhancement is in chloroform is much better. So we really need the calibration function here. Now we have here again the molar fraction of acetonitrile. And now because the enhancement of acetonitrile is much lower, of course, we underestimate the acetonitrile concentration in our sample. But we can still fit the data points with our curve and do the leave one out analysis. And then you end up with errors, which I'm not yet confident with, but I think most of the bad quantification relies on our stupid setup with that sudden expansion to the 3 millimeter and the bad flow dynamics that we have here. But at least now we can measure carbon-13 under continuous flow, which we couldn't do before, and we have found a way to quantify that. Okay, and with that, I'm already at the end. So I think that I have shown you that ODNP enables 1H and carbon-13 measurements in the fast flow regime, and we are starting to do a quantitative analysis in these samples. So far, we did it only in binary systems, and of course, we have to go to ternary and quaternary systems, and so on. We have definitely to improve our setup for carbon-13 ODNP, and then we will start to monitor simple dynamic processes until hopefully in the end we get everything fit to do real reaction monitoring. And with that, I want to thank the funders of this work and my team and you for your attention.
Thank you very much, Kerstin, for the very nice talk. So we do have time for questions. Well, first question for Kerstin, and then we'll go, maybe there are some more general questions. Krish, you had a question you want to ask?
Yes. Thank you, Patrick. Christian, that was fantastic. I'm really enjoyed this talk. I'm particularly impressed by the reproducibility aspect of this method. So the question I have is more like a blue sky, perhaps. Considering that your ODNP happens on both carbon and proton, is there a possibility of being able to do a one shot per increment 2Ds and exploit the Polish methodology? Kosminski published something about 6-7 years ago on time-resolved, non-uniformly sampled methodology to monitor reactions at an increment-to-increment level. And so essentially now we have opportunity to actually study kinetics time point at per increment level, and that time point can further be reduced by your ODNP methodology to make it per increment, per scan, kinetic time points, and study really fast reactions. I am really, really excited by this potential opportunity that it might introduce for real fast reactions. Thank you. There's no question. I just wanted to comment.
Okay, thank you so much.
Yeah, you want to answer, Chris? No, I mean it was more a comment.
No,
but,
no,
it's
it
okay.
wasn't a question. It was just a comment.
I just wanted to make sure that I give due credit to that idea. I don't know if Andreas Kerner is in the attendees list. This idea of fast reaction monitoring by 2D using time-resolved NUS was proposed several years ago by Andreas Kerner. So it's not my thing. I was impressed by his suggestion that was suggested in one of the SMASH meetings. That's why I remember. So due credit where it is due. So it is Andreas.
Thank you, Krish. I also had a question actually, Kerstin. So maybe my question is nonsense and maybe driven by my lack of knowledge of EPR. But you mentioned at the beginning that we would not get maximum enhancement, we would hope, because we can saturate only one EPR transition. Is that correct? Wouldn't there be a way to saturate more than one transition, for instance, by modulated microwaves or something like that?
Yeah. People worked on that actually, and we did already something. So if you use very high microwave power, which we are doing here, you get something like a microwave induced broadening of your EPR lines, and they start to overlap at the very bottom of the hyperfine line. So actually you can saturate more than one line by very high power microwave irradiation. Or what we are doing with that immobilization strategy is that now all our hyperfine lines fall into one very broad one, because we have now a very strong Heisenberg spin exchange, because the radicals are all very, very close to each other. And that gives rise to a homogeneous broadening of the EPR line. And therefore you can saturate more than one of the hyperfine lines. So actually we are still a little bit power limited, let's say. I think if we would have more microwave power, also the enhancements would be much better.
Thanks a lot. Other questions for Kirsten? Well, if not, then we have time for, I mean, general questions on hyperpolarization that some of you may have. And well, with Indrek and Kirsten, we are covering complementary aspects of hyperpolarization. So hopefully, if you have questions, one of us at least can answer.
Patrick, my question is based upon your presentation. How far are we with hyperpolarized, pure shift? I'm trying to pick the current exciting technology developments, combine them all to come up with a highly resolved. You did a 2D NMR quantitative. There are pure shift experiments that are coming up for resolution. Now we are addressing sensitivity. How far are we in going in metabolomics area?
So hyperpolarized pure shift NMR, there is one paper which has been done by Lucio Friedman. So, I mean, as usual, and it's the same actually with ultra-fast 2D NMR. We know that we can do ultra-fast 2D NMR with dissolution DNP. We've been showing that. Now there is a gap, and then people often forget about this gap between the development of a concept and talking about pure shift hyperpolarized NMR. It's really a concept at this stage. There is a gap between the concept and the application to real life, and that's the gaps that we are trying to fill. So it's really not simple, especially as soon as it comes to spatial encoding techniques. So I'm more specialized in ultra-fast NMR than pure shift NMR, but I suspect we will encounter the same kind of problems. As soon as it comes to spatially encoded techniques, there is an interplay between the residual sample motion after injection and the spatial encoding of the sample. So, for instance, developments that would require spatial encoding on an orthogonal axis in order that the spatial encoding is orthogonal to the residual sample flow are things that could help going along this direction. So if the question is how long are we from the application of these methods to metabolomics, well, I would not be surprised if it takes 10 years and 3 PhD theses. That's the kind of thing that can take time. Dissolution DNP itself was suggested 20 years ago. It took 10 years until someone started to apply it to natural abundant mixtures. And then it took another 10 years to be able to apply it to a small size metabolomic studies. And there are still questions here. So I would not be surprised if it takes another 10 years.
Okay, thank you. I might come back to you offline for some more
sure.
ideas.
I'll be happy.
May add a couple of details from the parahydrogen domain and pure shift.
Of course.
There was a paper from Manchester, I think 2 or 3 years ago, combining Sabre and pure shift. And well, it's strictly not pure shift, because I mean pure shift actually means a very specific combination of false program elements. But the NHPHIP team in the Radboud University in the Netherlands, they published late last year a similar to these 2D plots from NHPHIP that I showed, an advancement over these methods where each of those doublets will be a singlet, but they do not do the pure shift element. They more or less apply a homonuclear decoupling technique. But I mean, the end result is, similar. I'm not capable of saying whether better or not, but at least the pictures look beautiful.
Thank you. More questions. People can just turn their microphone on and ask.
And ask, of course. Yeah.
Yeah.
There are not so many now.
Anybody?
I'm sorry I had to arrive late, but I'll definitely be looking for these talks on YouTube. So if there are no more questions, though, I will end the meeting.
Thank you, Patrick, and thank you, Kristen and Indra.
Thanks a lot, everyone.
Excellent workshop. I loved it. Thanks.
Thanks very much, Chris. Bye-bye.
Bye-bye.